Are we ready to get started? All right.
I'm ready.
Okay. Thank you, everybody, for joining us today for a Friends from the Field webinar. These webinars are co-hosted by Blue Hill Heritage Trust and Island Heritage Trust. And if you're interested in learning about our archive of webinars from the past 3 or 4 years now, we have those on our YouTube channel and on our website, so feel free to check them out. All of our programming is free and open to the public, so if at any point you're interested in making a donation or contributing to our work, there's also information about that on our websites. And thank you if you already do, or if you just attend and take joy in these programs. We welcome everyone no matter what. And I'm going to pass it over to Noelle quickly for the tech, if that's okay, Noelle, and then I'll introduce Jordan.
Hi, everyone. I'm Noelle Ruth. I'm the Outreach Manager at Island Heritage Trust. Today, we're going to let Jordan share the presentation. If any questions come up while Jordan is sharing, feel free to drop those questions in the chat box. And at the end of Jordan's presentation, Lander and I will field those and hopefully get as many questions answered as we can.
Yeah, thank you, Noelle. I forgot to introduce myself, which I normally do, but I'm Lander and I'm the education coordinator for Blue Hill Heritage Trust. And so excited to be on here with Noelle and Jordan Chalfant, who is a naturalist who lives in Bar Harbor and studies seaweed. She's on the board of the Maine Natural History Observatory, and with a couple of her colleagues are writing a user-friendly photographic field guide to the seaweeds of Maine. And after spending 10 years working as a field biologist, doing botanical inventories and seabird monitoring on the coast of Maine, Jordan has taken on managing the Naturalist notebook, a nature and science bookstore in Seal Harbor, as her day job. So thank you so much, Jordan, for being here with us tonight. We're really excited to learn from you.
Excellent. Thank you guys for giving me this platform to share my love of seaweed with everyone. I'm just going to go ahead and share my screen so you guys can see my presentation. All right. Lander, does that look as it should?
It does. It looks great.
Excellent. All right. So yes, I'm extremely excited to discuss my favorite photosynthesizers, marine macroalgae. So in the next hour or so, I'm going to introduce you to the Seaweeds of Maine field guide and preview its contents by profiling a few species. I'll discuss terminology, life histories, ecology, and environmental concerns like introduced species and climate change. I hope that this introduction will enrich your experience in the intertidal zone and get you excited for our upcoming identification resource. Before I dig in, I'll give you guys just a little quick personal background and context for my involvement in this project. In 2008, I attended College of the Atlantic in Bar Harbor, where I earned a degree in human ecology with a focus on botany, ornithology, and scientific illustration. Post-college, I started working for a biological monitoring nonprofit called Maine Natural History Observatory, doing botanical inventories and field work for Director Glen Mittelhauser's various field guides. I worked quite a bit on plants at Baxter State Park, and I think Glen's most well-known field guide is probably Plants of Acadia National Park. Post-college, I also worked as a contract field biologist for Maine Coastal Islands National Wildlife Refuge. I monitored breeding seabirds on islands off the coast of Maine. I mixed that with some guiding in Acadia, state gardening, and now, as Lander mentioned, my day job is managing a nature and science bookstore in Seal Harbor called the Naturalist Notebook. In addition to plants and birds, I became interested in seaweed while I was in college. I took a winter coastal ecology class, and part of that class involved making a collection of seaweed vouchers, which very rapidly evolved into an obsession with walking the shoreline and looking for interesting washed up seaweeds and making art with them. I loved the science and art crossover. Eventually, I started noticing patterns and becoming aware of the incredible diversity of sea plants on the coast of Maine. And I was really frustrated with the lack of user-friendly field guides and identification resources for seaweed. I expected that I would be able to put a scientific name to the species I was encountering, just like I did with plants. And that's really not the case. Um, so these are the identification resources that are currently available for the Northwest Atlantic to give you guys an idea of what's out there right now. These are the ones that go beyond the 10 or so most common species of marine macroalgae. One of my favorites is Martine's Illustrated Key to Seaweeds of New England. The key in it is really great. She cuts out a lot of the excessively complex terminology and mostly focuses on macro characters, things you can see without a microscope. And she even supplements with some helpful, albeit kind of grainy, black and white photos and a few line drawings. Unfortunately, the key includes only around half or so of the species found in Maine, and the names, ranges, and taxonomy are out of date. Plus, the book is out of print, and the only copies I can find are over $100 online. The Northeast Algal Society keys are excellent, useful keys, but again, they're kind of out of date. They have sparse visual aids, and this key is not easy to come by as well. I couldn't find any copies online last time that I looked. Dr. Gary Saunders of the University of New Brunswick, who's one of the foremost phycologists or people who study seaweed in this region, is working on a new edition, but it's still a ways away, and it will be quite technical when it does come out. Gary also has a website that I recommend, seaweedcanada.wordpress.com. It's a truly magnificent, albeit in progress, resource for taxonomy and identification of seaweeds. If you know what you're looking for, the online resource doesn't have keys, so you kind of have to already have an idea of what you're trying to investigate in order to get to the right information. Algae base is worth noting that it is sort of where all the seaweed researchers I know go for the most up-to-date information on taxonomy changes. They have vetted photographs and other references on there. So if you've got the seaweed bug, that's a great authoritative online resource to check out. Seaweeds of the Northwest Atlantic by Matheson and Dawes is an authoritative tome. It was just released in 2017, and it's an essential resource for species descriptions and taxonomy to date. But after years of frustration struggling to use the keys in this book, my colleague confirmed my suspicion that some of the keys simply don't work. It's also very terminology heavy. You have to majorly grow your vocabulary to make sense of it, and it relies almost exclusively on microscopic characters like cell shape and size and number of plastids and whatnot. And while there are no photographic field guides to the seaweeds of Maine, or even the Northwest Atlantic in general, there's a wonderful field guide to the seaweeds of Britain and Ireland by Bunker et al., which has maybe around like a 50% overlap with our flora, so not enough to be a standalone resource, but still helpful. So I asked Glen Mittelhauser if he would write a user-friendly photographic field guide, and he responded that I should write a field guide, and we compromised by agreeing to write one together. I think there is great need for such a book, not just for classification nerds and hobbyists like me, but also as a tool for resource management and preservation. I like to envision a world where seaweed diversity and ecology is understood in such a way that it can be used to evaluate the health of our intertidal and subtidal systems as a tool to create protective legislation. Seaweeds have the same capabilities as plants to be used as indicator species. And in the words of my colleague, Dr. Jessica Moulin, who is a phycologist at Maine Maritime Academy. Seaweeds are experiencing a renaissance. They are gaining popularity as health foods. In less than 20 years, the number of businesses that wild harvest or grow seaweed has jumped from fewer than 10 to more than 30. The Department of Marine Resources reports that commercial harvest of rockweed has more than tripled over the same period of time, driven in large part by its use in soil remediation. I think that being able to identify seaweed is an important first step to good stewardship. You might be kind of wondering how I think I'm going to write a useful and accurate book when I'm mostly self-taught and have a BA in human ecology. And that's a valid question. But I think actually that my amateur background is in some ways an asset. I still remember the bridges that I had to build for myself as I figured out new terminology and translated it into familiar terms. I know which couplets work and which are useless to beginners and existing marine macroalgal keys, and what macro characters are being overlooked in favor of cellular features just because of tradition. I'm also really focused on the illustrations for our guide, and I've been developing a few techniques for photographing seaweed that I'll touch upon in a little bit here. And luckily, I have an extremely experienced field guide writer and an actual phycologist on my field guide writing team. In addition to authoring more than 5 field guides, Glen Mittelhauser has coordinated some of the most comprehensive biological monitoring projects ever conducted in the state of Maine, including the Maine Seabird Atlas and the Maine Breeding Bird Atlas. Um, He's also working on a saw-whet owl and a nightjar monitoring project, and he does dozens of botanical inventories, including a complete survey of Landing Garden Preserve on Mount Pleasant Island, which I'm really excited about. Um, During the pandemic, Glenn and I took 2 week-long online macroalgae classes from Dr. Amanda Savoie through Eagle Hill Institute in Steuben, Maine. Dr. Gary Saunders was Amanda's PhD advisor, and they specialize in using DNA sequencing to delineate seaweed species, and they have published a ton of papers about it. For her PhD, Amanda restructured we call it Polysiphonia sensuato. So it used to be one genus, now it's a whole bunch of different groups in the Polysiphoniae. It's an extremely challenging group of red seaweeds that have been stumping taxonomists for centuries. Each alga on this slide is a different species. So as you can see, without magnification, they can be quite difficult to distinguish. Amanda is a researcher and phycologist at the Canadian Museum of Nature. She's also in charge of their Arctic research program and is learning tons of novel and interesting things about how post-glaciation seaweeds have migrated from the North Pacific and Arctic Ocean to the Northwest Atlantic, as well as from the Northeast to the Northwest Atlantic. Her work is crucial for documenting the effects of climate change as seaweed species in the rapidly changing Arctic may be disappearing before they've even been documented. In May of 2022, Amanda was slated to teach an in-person macroalgae class at Eagle Hill, but only Glenn and I signed up and the class was canceled. So instead, the 3 of us did an informal and spontaneous week of seaweed collecting, identifying, photographing, and preparing vouchers and DNA samples. It was one of the most intense and blissful experiences of my life. The tides were in the early morning, so I was getting up at like 3:30 in the morning and then working until 10 p.m. at night. And even as the tides got later in the morning, we kept going further and further afield, like up to Lubec, Maine. But I wasn't even tired because I was having so much fun. And it was so amazing to finally have someone who is equally as obsessed with seaweed as I am to geek out with and to learn from. And Amanda had been cooped up at in Ottawa for the entire pandemic, and she was just as delighted to be exploring the coast of Maine. So at the end of the week, Amanda signed on to co-author the guide with us. Yay! I think we're a great team because Amanda is a thoroughly trained phycologist who is on the leading edge of seaweed taxonomy work. Glenn is an accomplished botanist with a ton of experience writing field guides, and I am very determined. All right. So I'm learning the first step to writing a field guide is figuring out what species should be included. There's unfortunately no official species list or comprehensive database of historic to present seaweed records in the state of Maine. As such, we're building a relational database that includes herbarium vouchers, published papers, and unpublished species lists, and we're incorporating partial databases like the Macroalgal Herbarium Consortium and the Barcode of Life data system. So far, we've researched Washington and Hancock counties, adding 13,952 herbarium vouchers, 5,459 published location records, and 326 unpublished observations documenting 322 seaweed taxa in eastern Maine, as well as their distribution and temporal patterns from this region. Eventually, our database will be public access, but first we estimate we have around 30,000 or so records to go in southern Maine, and that our final number of species will be close to 400. Glen designed our database to track nomenclature changes. This is important because there have been and continues to be major upheavals in the taxonomy of seaweeds because of new DNA-based evidence, which is resulting in the creation or modification of many species. In addition to developing a species list from historic records, we're doing a lot of field work to update species lists. We're also photographing species in situ at low tide, collecting specimens that require microscopy work for identification. And in the case of cryptic species that cannot be identified because of ongoing taxonomy confusion or lack of reliable characters or because of morphological plasticity or insufficient data because of recent discovery. We're taking DNA samples, which Amanda can sequence at the museum. I think visual depictions are crucial to making seaweeds accessible to people. Photographing seaweeds is challenging on multiple levels. Using the common red alga Polysiphonia stricta as an example, out of water, most are amorphous lumps or little slicks on the rocks. And on the one hand, that's how most of our guide users will be encountering them, so it's important to show. On the other hand, you usually can't distinguish identifying characters unless the seaweed is submerged. In situ underwater photography is very difficult because of water movement and sediment obscuring the image, but we do try. And we also try to get field photos of the habitat, since where in the intertidal zone and on what kind of shore a seaweed is found is often an important identifying character. We then take samples to a lab setup where I use plexiglass trays of seawater and a light pad to capture a three-dimensional profile. We also have a fantastic Olympus CX43 microscope with a camera so we can document microscopic characters. In the field guide, we'll rely almost entirely on macro characters, but in some cases, microscopy is a good way to get very clear images of things that you can just distinguish with a hand lens. And we'll also include scanned pressed specimens when it's appropriate. So here's an example of what a species page will look like in our guide. This is Desmorestia viridis. We get straight to the important stuff by beginning with the combination of characters that distinguish this species from the rest of Maine's flora. This is followed by a full description comprised of our own observations plus a synthesis of the existing literature. As we complete the database, we'll be able to give distribution and phenology information and a commonality designation for each species. As mentioned, habitat, including depth and the intertidal or subtidal zones, and substrate and wave exposure are important identification factors that will go in the distribution and habitat section. We also include a paragraph where we specifically address lookalikes. And we put in any comments that we have about interesting factoids. For example, Desmarostia viridis is the only genus of seaweed found in Maine that could be dangerous to eat. It has vacuoles containing sulfuric acid with a pH of 1. It's thought to be an anti-herbivory compound that can damage the teeth of sea urchins. I learned early in my seaweed collecting endeavors that you absolutely must keep this seaweed in a separate bag from the rest of your collection, because when the seaweed is stressed by being removed from the ocean, it releases all of its sulfuric acid and it'll melt itself and any other seaweed it comes in contact with to goo. We also include a paragraph where we specifically go into the life histories of the seaweed. I'm not going to go into great depth on seaweed reproduction either in the guide or in this presentation because frankly it's a huge headache. Reproductive strategies and life cycles vary tremendously from species to species and involve 2 to 3 phases. I'm hopeful that Amanda will tackle this part of the guide. Common names are even less relevant in seaweeds than they are in plants. Hardly anyone uses them, and every region and demographic has its own name, so there's even less standardization than there are with plants. This is particularly frustrating with seaweeds because there's so much upheaval in the scientific nomenclature as well. And I don't have a solution for this really annoying problem. Uh, but I strongly recommend that if you're interested in learning seaweeds, you do so by learning their Latin names rather than relying on common names that haven't been standardized. Currently, we've finished photographing and describing about 30 species of seaweed, with twice as many in various stages of completion. And although this is a small fraction of the few hundred species found in the state, it's a larger percentage of the seaweeds that will be included in the guide, as we'll be focusing on those that can be identified using macro characters. I don't have exact figures yet, but I'd say as many as half or so of the seaweeds of Maine are indistinguishable without microscopy or DNA work, so we'll only mention those generally in genus or in group descriptions. So full disclosure, I had a little bit of a hard time with the flow of this presentation, and I decided to talk about the guide first and then pivot and talk about seaweed generally. Um, and so halfway through the talk, I'm going to go way back and actually define the group that I've been talking about this whole time. And then we'll get into a few species profiles after that. So what is seaweed? Seaweed is synonymous with marine macroalgae. Seaweed is algae found in the ocean that can be seen with the naked eye. Generally speaking, seaweed is comprised of 3 distantly related phyla: Rhodophyta, informally known as red algae; Chlorophyta, which includes green algae; and Phaeophyta, brown algae. There are some seaweeds that do not fall into these 3 groups, and there's also members of these groups that are not seaweeds. So it's not an exact division, so to speak. There's so much top-down taxonomic revision going on. I'm not really comfortable with providing figures for the number of species in each of these groups. However, these are algae basis tallies to give you a sense of the general numbers and ratios globally. Algae base includes freshwater algae, so it's worth noting that about 95% or so of the Rhodophyta and Phaeophyta are marine, but only 10% of Chlorophyta is marine. The ratios in the Northwest Atlantic are similar to global statistics. Dr. Gary Saunders recently estimated that there are 120 greens, 185 browns, and 575 reds in Canada's flora, though he qualifies his numbers by stating that his lab is biased towards red algae, and Amanda and I sort of are as well. And I think that we're all overlooking a significant number of cryptic green species, so that number is probably actually larger for marine Chlorophyta. So this graphic shows how green algae are, oh, where's my little, oops, shoot, hang on a second, I'm gonna go back. Um, Let me just, I want to make my little cursor the, oh wow. All right, you can probably see with my cursor, but over here, um, you can see that green algae are the predecessor of land plants, of vascular plants, and red algae diverged way earlier than vascular plants and green algae, making green seaweeds more closely related to plants than to red seaweeds. And as a comparison over here in this pink box, you can see that like fungi and animals are more closely related to each other than green algae and red algae are to each other. And brown algae are all the way over here in a separate group and are more closely related to diatoms than they are to green or to red algae. They're also related to oomycetes. I don't know if you guys know about these things. I've just recently learned about them. It includes a bunch of plant pathogens like tomato blight and mildew, which scientists until quite recently thought were fungi. So all of this is mind-boggling to me because sometimes only the most minute morphological differences separate these groups. And informal use of color names makes things worse because red algae are often brown or greenish, and brown algae can be green, and green algae can be brown. So here's like, if I were just like looking at some seaweed, how I might categorize these organisms here, but no, all wrong. This is not brown algae, it's actually a red alga, Porphyra. These are colonial diatoms. This is another red alga in the Polysiphonia group. Corallina officinalis is a calcified red algae. And a lot of brown algae, like Desmarastia viridis, oops, there's supposed to be an I right there, and Pileella littoralis turn green after they've been pressed, so that can be confusing too. And crustose coralline algae, kind of like rocks, but they're actually algae. So what unites seaweed is less their evolutionary history or appearance and more where they're found and how they function within marine ecosystems. Oops, I was gonna, yeah, we can go on to that. So one of the other things that defines seaweed is that they are almost all photosynthesize and they occur in a band from the high tide line down to 50 or so meters, give or take, with some exceptional species that are found even deeper, like that rock-looking pink alga I showed you guys. Um, they provide habitat and structure for other marine life, and they serve much the same role as plants do in terrestrial environments. And so since we've established that seaweed is a polyphyletic grouping, we're gonna now take a closer look at the three primary groups. And since most identification resources start by dividing seaweed keys into red, green, and brown, you're gonna be in for a lot of frustration if you can't place your mystery species into the correct phylum. And there aren't a lot of characteristics unifying these groups that are useful in field identification, but I'm going to touch on them anyways. We'll start with the red algae because they're my favorite. They have the most complex life histories and they're the most diverse and intricate in appearance, so it's hard not to be attracted to them. The defining features of rhodophytes are that they lack flagella and centrioles, and they have plastids enveloped in two membranes because of a process called endosymbiosis, which if you don't know about it, you should Google it later because it's totally whack. Um, the loss of flagella is what's thought to have spurred the development of a triphasic life cycle, which is also a defining feature of this group. They have a gametophytic and two sporophytic generations. Red algae also contain phycobilin accessory pigments, which are responsible for their color. And almost all red algae have pit connections between their cells, which makes them a little bit stretchy. This character is actually super helpful when you're trying to decide whether you have a brownish red alga or a reddish brown alga. If it's elastic when you pull on it, it's probably a rhodophyte. The unifying features of the chlorophyta include the presence of flagella and the lack of phycobilin accessory pigments that would mask their green color. Green seaweeds are generally structurally quite simple, which makes them very challenging to identify and characterize. Some newer classifications have shifted Phaeophyta down to class Phaeophyceae and named the restructured phylum Orchophyta, which is defined by minute features such as two flagella on their motile cells. One has distinctive hairs and also the presence of carotenoids, the pigments found in brown algae. And typically brown algae are pretty obviously brown, but it's good to keep in mind that some, like Desmarastia viridis that we saw before, and like Colponia peregrina is another example, they turn greenish when they deteriorate or after they're pressed. The same is true for red algae, which often turn white or green when their phycobilin pigments are damaged. Oops. All right, so I thought I had another slide in there. Um, with the remainder of our time, I would like to profile a few representative species that you're likely to encounter on the coast of Maine. And these species also serve to introduce more subjects in the realm of marine macroalgae, like edibility, climate change, ecological roles, life histories, morphology, and terminology. I think that when people envision seaweed, usually they're picturing members of the order Laminariales, or kelp. The most macroscopic of all the algae, Pacific species, Macrocystis pyrifera, or giant kelp, can grow to 45 meters in length at a rate of up to 60 centimeters a day. All this growth necessitates tissue differentiation, of which kelps exhibit a bit more than most seaweeds. Here in this cross-section, you can see they have a myristoderm, cortical, and medullary layers. Maine's flora includes 5 species of kelp, give or take. The largest and most abundant of which is Saccharina latissima, or sugar kelp. If you studied seaweeds in the past, you probably know this species by its former name, Laminaria saccharinea. In winter and spring, it can grow 2 inches a day and can reach up to 6 meters in length. It's one of the most widely distributed species in the North Atlantic. Um, So sugar kelp, like most laminariales, grows subtidally and is only exposed during the lowest tides. If you were to compare seaweed communities to plant communities, kelp forests would share a lot of characteristics with old growth forests. Kelps are perennial, surviving up to around 5 years or so, which is long lived for a seaweed, and they provide relatively stable, sheltered environments. While they're canopy forming and dominate the substrate, there are plenty of understory seaweeds in kelp forests and a great diversity of micro and macroalgal epiphytes found on Saccharina latissima. So it sort of creates structure in the intertidal zone and provides habitat. This is also a great species to use as an example of basic seaweed anatomy. These are some of the terms that you'll encounter if you ever try to identify seaweed using any sort of written resource. The basal structure on a seaweed that anchors it to the substrate is called a holdfast, which makes sense. Sugar kelp's holdfast is branched and rooty. It's extremely strong. It can withstand multi-ton forces of waves and the associated drag that goes with that. Um, Holdfasts differ from roots in plants. It's worth noting. In plants, root tissue is specialized for nutrient and moisture uptake. In seaweed, those processes take place throughout the alga. Holdfasts vary morphologically, and it can be important in identification. So these are just some other examples of seaweed holdfasts that show some of the common types. They can be rhizoidal filaments or stoloniferous. They can be prostrate. They can have little tiny discoid holdfasts. Um, All right, and the next part up from the holdfast is the stipe, which is similar to a stem. It refers to the space between the holdfast and the first blade or first branch. When the thallus flattens out, it's called a blade, and sugar kelp growth occurs right here, where the stipe transitions to the blade. In many other seaweeds, growth is apical, happening at the most distal tips of the plant. Um, how a seaweed grows is often important in some of the more technical identification processes. Um, the The flat blade provides a lot of surface area for photosynthesis, kind of like a leaf. Seaweed has to strike a balance between maximizing photosynthesis and minimizing drag. Wave force has a major impact on seaweed morphology. If you're a seaweed that's too big or too rigid, you're going to get ripped off of the substrate. For this reason, sugar kelp growing in wave-exposed energetic waters is kind of narrow and strap-shaped, very linear. In calmer waters, sugar kelp is broad with ruffled edges like this pictured specimen. That increases the surface area and therefore its ability to photosynthesize. Kelp has been commercially important for hundreds of years. In Europe in the 1700s, prior to the discovery of mineral deposits, it was harvested and burned for potash, sodium carbonate, and iodine. In the 1800s, sugar kelp was popular for its alginate content. Alginates have water-holding, gelling, emulsifying, and stabilizing properties that can be put to use in food manufacturing, in medicine, paints, and fibers. Today, sugar kelp is primarily harvested for food. As more and more people are attracted to its health benefits, it contains numerous vitamins and minerals, as well as iodine and antioxidants. And all of my seaweed edibility and harvest information should be taken with a grain of sea salt because I am a taxonomist. I focus on seaweed classification. So I'm able to provide you guys with some superficial information here, but I'm probably not the best for any of the deep digging sort of nuances of seaweed harvesting and things like that. Double-area Ramentaceae is this species in the center here. It's far less abundant than our kelp species, and it flourishes on the most exposed coastline with incessant pounding from waves. Maine is the southern end of its range, and it is endemic to the Northwest Atlantic. There's concern that warming waters in the Gulf of Maine will contract the range of this cold water-loving species. And there are similar concerns for sugar kelp and Palmaria palmata or dulse. Devillaria is a red alga. Its branches are pretty obviously red, but when it's denuded and only the primary axis is visible, it can be tricky to place it in the correct phyla. Devillaria is a perennial. Its branches tend to die back in summer, but it can regrow from its tough main axis. This species is a great one to use as an example of the incredible morphological plasticity of seaweeds. Seaweeds rarely adhere to the rules that plants more reliably follow, such as branching patterns, size, shape, color, phenology. You name the rule, seaweeds are right there breaking it. I think the most repeated word in our field guide is going to be usually, because the only always with seaweeds is that you always have to apply a qualifier. There are always exceptions to whatever you're trying to say about seaweed. But like other seaweeds, Devillaria's appearance depends a lot on environmental conditions. So even this has unpredictable effects. But generally speaking, as I mentioned, seaweeds have a broader form in calm waters as they try to make up for the lack of upwelling in nutrients in the water by increasing their surface area. And they have more streamlined or compact forms when they're subjected to strong currents and wave action. You can see that the Devilaria in these pictures here ranges from simple blackish sticks that are just a few centimeters high to proliferous short branches to just a few long inflated branches. Devilaria with long, wide branches can be difficult to distinguish from Dumontia contorta, an introduced seaweed from Europe. I'm usually able to tell them apart because Dumontia's main axis usually is sort of the same consistency as its branches compared to Devilaria, which has a really tough blackened main axis. Also, Devilaria's branches are rarely twisted, while Dumontia's often are, and they are found in different places in the intertidal zone. Devilaria is very low intertidal to subtidal. Sometimes I see it in deep tide pools further up, but Dumontia is upper intertidal. However, their ranges do overlap, so you, as with most seaweeds, have to use a combination of characters to suss out a species identification between these two. All right, so Dumontia is one of around 14 introduced seaweeds in Maine's flora. As with plants, introduced seaweeds vary in their environmental impact. Some are quite rare, some are hopelessly proliferous, they have the ability to alter natural communities and cause economic harm. Some have been here so long that their introduction is inferred by genetic evidence rather than by first documentation. And some are so recently introduced that we don't yet know how they're going to affect Maine's biota. While Dumontia contorta is the introduced species that I encounter at the greatest frequency and in the greatest abundance, I haven't really observed it displacing or excluding other species of macroalgae or causing any obvious ecosystem or economic harm. Dumontia is tolerant of an exceptional range of conditions, and it sort of seems to utilize spaces that are inhospitable to other seaweeds. I find it in mudflats, in freshwater influenced outlets, on exposed coasts, in energetic or quiet waters, in high intertidal pools. It grows on rocks, pebbles, or shells. It's most abundant in late winter and spring, but it can be found year-round. Typically, the annual upright part of the plant dies in the summer and regrows from a perennial crustose base. In contrast to Dumontia's relatively unobtrusive habit, the most invasive of seaweeds introduced species, in my opinion, is Dasyphonia japonica. This species is native to the Asian Pacific. It was introduced to France in 1984, and it spread rapidly throughout the Northeast Atlantic. From there, it was transferred to the Northwest Atlantic, probably via ballast water in ships, and it was first recorded in Fort Wetherill, Rhode Island in 2007. In 2012, Dasyphonia japonica was recorded in Cape Elizabeth, Maine, and by that time, it had become a nuisance species in southern New England. It fouls up fishing traps and washes ashore in giant mounds that release noxious gases when it decomposes. Also in 2012, before any records from mid-coast or down east Maine, Dasyphonia was recorded for the first time in Nova Scotia by Amanda and Gary, who hypothesized that maybe recreational boating facilitated the rapid jump in distribution. I began regularly visiting the Seal Harbor Beach on Mount Desert Island to collect washed up seaweeds around 2011. And it wasn't until 2017 that I recorded Dasyphonia japonica for the first time. Now in 2023, with the exception of rockweed, sugar kelp, Ceramium virgatum, this is the species I encounter most in drift at this location and in nearby Bracy Cove. I've also encountered Dasyphonia japonica at over 70% of the 30 plus locations where I've collected seaweed in down east Maine, ranging from estuaries to over 30 kilometers offshore on Maine's most distal island, Mount Desert Rock. Because this is primarily a subtidal species, I have to rely on wash-ups and occasional intertidal specimens to indicate its presence. However, I have received reports from divers that this species has recently become abundant in Frenchman Bay. Of all of the introduced seaweeds I've read about and observed, this one concerns me the most, given its capacity to alter species composition and ecology. So, for example, in a study conducted in the Gulf of Maine at Isle of Shoals, subtidal surveys that were predominantly kelp communities in the 1970s and 80s were replaced by the introduced codium species in 1990, which then in turn was replaced by Dasyphonia japonica as the overwhelmingly dominant species in the 2010s. Seaweed japonica is tolerant of a wide range of environmental conditions, and studies have found that it is more effective at nitrogen uptake than comparable native species, which compounds the negative effects of eutrophication. Dasyphonia japonica's methods of proliferation are quite overwhelming. It's not known to reproduce sexually in the Northwest Atlantic. It reproduces by small vegetative fragments that reattach to substrate and grow rapidly. This species is pretty easy to recognize. When Dasyphonia japonica is stressed, it turns remarkably bright orange to pinkish in color. You can spot it from a mile off. Healthy specimens are darker red and may be confused with a number of other branched filamentous red algae. But you can look for the following combination of characters which are distinctive. There are corticating cells on the main axes. So this is a main axis right here, this thickest part of the seaweed. And you can see that there are little small cells scattered around there. Those are corticating cells that are on the outside of the inner cell arrangement. And then it has visible polysiphonous cell arrangements with 4 to 5 paracentral cells in the final orders of branching. So right over here, you can see examples of that polysiphonous cell arrangement. That word refers to when there's a central cell that's surrounded by a ring of other cells that are all the same length. That's a polysiphonous arrangement. Here's a cross section in the bottom left of the screen that shows that cellular arrangement. And so when you're looking at it from the side view here, you're seeing 1, 2, 3 of those 4 paracentral cells. And then the branchlets are uniseriate, which means that they're a single chain of cells. Each one of these little pink logs is a cell. The branches are closely spaced and they appear somewhat tufted at the branch tips, and the main axes are usually less than 1 millimeter in diameter, and they taper towards the tips. And it really actually only takes like a 10x or higher hand lens to see these features. You don't have to have a microscope. The next species I'll profile is Ascophyllum nodosum, also known as knotted rack or asco. It's the only member of its genus, but when referred to generally with other species in the Fucaceae family, it's called rockweed. Along with the other rockweed species, it creates a clear band of thick, dark to light ochre brown rack in the uppermost part of the intertidal zone. Rockweed is thick and robust and adapted to tolerate long periods of exposure while the tide is out. Asco is the most abundant seaweed in Maine. It grows on rocks in estuaries and on open and exposed coastline, quite a range of habitats. It's attached by a nodular discoid holdfast. Its main axis and primary branches have large, single, irregularly expanded bladders. You can see them right here. These are filled with gas, and when the tide is in, they add buoyancy, which brings the seaweed closer to the sunlight at the water's surface. You can age Ascophyllum by counting the bladders on the main axis, as it pretty reliably adds 1 bladder per year. Asco is one of the oldest living seaweeds in the Northwest Atlantic. It can reach up to around 15 years old. And just like plants, seaweeds can be annual or biennial or perennial, but they're less obligately so. As usual, they don't always follow the rules. They can do whatever they want. It has branchlets in the shape of little clubs, which are converted to or replaced by yellowish receptacles containing reproductive materials in spring and summer. Some people like to eat these and call them sea grapes. ASCO is loaded with receptacles right now, guys. This is like the time. So if you want to try sea grapes, get out there in the intertidal. ASCO has much the same historic uses as sugar kelp. It's currently harvested primarily as fertilizer and as supplements in livestock feed. Although ascophyllum is harvested more and more heavily and unregulated harvesting is an extremely contentious issue, there isn't that much evidence that at the current levels of harvest that there's significant ecosystem harm. The most recent study that I read indicates that ascophyllum regrows quite readily as long as it's cut at least 6 inches above the holdfast. And I qualify those statements by reminding folks that I'm a taxonomist. I'm focused on classification, and I'm not an expert on resource management or ecology. But that is definitely something that's at the forefront of people's minds these days is seaweed harvest and making sure to do that responsibly. So I encourage everyone to educate themselves. Ascophyllum on exposed coast is frequently subjected to the obligate hemiparasitic red alga vertebrata lanosa. It was considered an epiphyte until a recent study indicated that it actually penetrates the tissue of ascophyllum and derives some nutrients from it, in addition to doing its own photosynthesizing. Vertebrata lanosa is only found on Asco and very rarely on other fucoids, but it never grows on rock. This is the third polysiphonous seaweed that we've looked at, the first being Polysiphonia stricta and the second Dasyphonia japonica, both of which have 4 paracentral cells surrounding a central cell. Vertebrata lanosa has 20 to 24 paracentral cells. It looks quite beautiful in cross-section. There are around 24 or so seaweeds in Maine's flora that have polysiphonous cells. I've found some excellent, seemingly reliable macro characters that should allow us to key these species without microscopy work in the field guide. Vertebrata is distinct amongst other polysiphonous algae for its structural integrity. If you dry it off just a little bit, it puffs up and becomes very three-dimensional, like in this image here. And it's sort of the only one that has structure quite like that. That's all the species that I have time for, but in case your interest is piqued in seaweed and you'd like to learn more, here are several ways you can do that. You can visit Maine Natural History Observatory's website, mainenaturalhistory.org/seaweeds. You can find more information about the project, seaweed identification infographics, and a free-to-download guide to common and sometimes overlooked seaweeds of eastern Maine, which is sort of a mini guide that we put together with a small grant. It's a great preview of what the full guide will entail. If you're into social media, you can follow us on Facebook and Instagram at Seaweeds of Maine, and I post lots of photographs and species descriptions and previews of the guide on there. May 21st through the 27th, our field guide co-author Amanda Savoie will be teaching a week-long in-person seaweed class at Eagle Hill Institute in Steuben, Maine, entitled Marine Macroalgae Ecology, Identification, Distribution, and Importance. Amanda is so knowledgeable and approachable and fun. I strongly recommend going if you possibly can. And once again, we're not sure we're going to have enough participants for the class to proceed. So if you think you might want to attend, I'd recommend signing up as soon as possible so that the class isn't canceled. On June 10th and 11th, I'll be leading seaweed workshops at the Naturalist Notebook, the nature and science bookstore that I manage in Seal Harbor on Mount Desert Island. The first one is focused on seaweed collecting and identifying. And the second one is all about the art of seaweed pressing, and you'll get to try that out in the workshop. Information on both those events can be found at thenaturalistnotebook.com/events. Finally, if you would like to help us make the Seaweeds of Maine field guide a reality, these are a few areas in which we could use some help. We would love to have volunteers help us by photographing seaweeds in the intertidal zone. You can email us for an instruction manual on taking useful photos. We're also looking for recommendations for public access coastline where we can conduct surveys and collect samples. Specifically, we're looking for exposed rocky coast. Those are places that get a lot of wave action and will have greater seaweed diversity. We're also looking for seaweed records to add to our database. So if you know of any surveys that are likely to have been missed by us because they aren't published or public access, please let us know. And we are in search of funding. Right now, the publication is a solid 4 years or so away. We've received a couple of small grants that have helped fund part of the database and the mini guide, but currently we're working on the guide in our free time. And it would be great to have the financial backing to actually really focus on producing the field guide. So please reach out to us if you have any suggestions of places where we should apply for funding. And eventually we'll be looking for volunteers to try out our keys and to field test the guide. If you have input or you would like more information, you can email me, Jordan, or Glenn. Our email addresses are on the screen there. And thank you guys all for your time and interest. I would love to take a stab at answering any questions.
Thank you so much, Jordan. What a rich
Thank you, Jordan.
learning experience. We do have some questions. Maybe we can get started with those.
Let's do it.
Let's see, I have to pull it up. Do you have it in front of you yet, Noelle? I'm just getting it myself.
I think the very first one that came in was a question about one of the first photos you shared, Jordan. It was there was like a whole mix. I mean, all your photos are like a mix of seaweed. How do I describe it?
Let's see, well, let's just go back to the
It was like one of the very
beginning
first
of the-
pictures, and the question is the location. Where is this location?
Let's see here. Hang on. Let me see if I can share my screen again. So this is. Does it say? I can't look at the
It was
comments at the moment.
it was before that one. I think it was like one of the very first of your introduction.
This one?
That one. Yep.
I believe. Let me think. I don't remember off the top of my head, so I'm going to stop sharing my screen and then go through my photos. And can you guys fire me another question while I'm looking?
Yeah. Lander, do you have the next one?
I do. So someone else early on had asked, what database software are you using for your relational database?
Is Glenn still here? I don't remember what it's called. Hang on a second. I can figure that one out too. Okay, so the this is what I thought and why I hesitated. So the first photograph is taken on a private estate that the landowner asked me not to share the exact location, but it's in Bar Harbor near the shore path. And then, so the seaweed database. It's we're using Microsoft Access database platform.
Okay, the next question I think is if someone who is allergic to iodine, shellfish, could eating raw kelp be bad? This person is a kayak guide, so that's probably a good thing
I
to
would
know.
imagine, but again, I don't have a ton of knowledge about that. There's a ton of iodine in Saccharina latissima, so I would say that that is a valid concern, and I can recommend to you Maine Coast Sea Vegetables is a nearby wild harvest seaweed group. Shep is the guy at the helm, and those guys know more about seaweed harvesting than anybody else I know. And Maine Sea Vegetables website has an insane amount of information about seaweed biology and harvest history and things like that. So those would be the guys I'd recommend going to for a more specific answer to that question.
Well, that's great. Another question is, can you tell us how you keep your collected groups of seaweeds fresh until you get them organized to photograph them?
Oh yeah, good question. So I when we're out in the intertidal zone, we carry with us stasher bags. Used to people primarily used like Ziploc bags, but number one, you only get a couple uses out of those before you pop a hole in them, and number two, they leak quite easily. So stasher bags are like those silicone reusable bags, and I'm a huge fan of them. They need to sponsor us because I keep telling people about them. But you put your seaweeds in a stasher bag along with as much seawater as you can fit in there with them. And then we take them back and store them in a refrigerator. Definitely you don't want it to be like near freezing temperature, but like 40 degrees or so will keep seaweeds fresh in a bag of seawater for, depending on the species, a week or so.
Awesome. Is Hemanthalia elongata / sea spaghetti found in Maine? I feel like I'm seeing some washed up, but didn't think it was.
Yeah, I'm just looking for that question so that I can see the spelling of that name.
It was sent at 4:45 p.m.
Oh, dope. Thank you. I'm not familiar with that species. I have not seen it, and I'm guessing by the name of it that it probably looks a lot like Demontia contorta. I'm gonna look that up while you guys shoot another question at me.
Okay, cool. What is the best kind of environment to find sugar kelp at low tide?
Exposed coasts, so like places that get a lot of wave action. You can make it easier on yourself by going to places that have a lot of variation in the topography, so that you can get to spots that are lower down while staying out of the water yourself. So yeah, that's about as specific as I can be. Exposed coast is usually better. You want some wave action, but as I mentioned, Saccharina luteissima is one of the most widely distributed northern Northwest Atlantic species, and can even be found in calmer waters as well. Yeah, I think that the key is just going out at a really low tide. So the kelps, as I mentioned, are typically only exposed and accessible, when the tide is like 0 or less. And you can find out exactly what the tide is and what time the tide is by consulting a tide chart. If you just Google search tide chart Blue Hill or something like that, it'll come up, and you can plan your expedition around when the water's low enough to gain access to that species. So I'm looking at pictures of Hemanthalia elongata, and I can say that that is not a species that we have here in the Northwest Atlantic. It looks like it is present in kind of the same environment, but over in the Northeast Atlantic. And I'm not sure what you would be confusing that with. Maybe Fucus disticus, and other species in the Fucus genus. But I would totally encourage anybody who has seaweed mysteries to send me photos. I love seaweed mysteries. My hobby is to go on iNaturalist and like correct people's identification because it's so fun. And with the qualification that sometimes these species have very microscopic and very specific characters that delineate them. So depending on the quality of your photographs and descriptions, I might only be able to get so far.
Wonderful. Thank you for being so thorough with your response. That's really kind. We have several compliments here about the design and how well you provided information. Beautiful and fascinating presentation. And then another question is, what is the difference between rocks that are seen covered with seaweed and those right next to them that have no seaweed on them?
That's an excellent mystery that I wonder about sometimes too. And I don't really have an answer for you, except that depending on the size of the rock, sometimes in winter storms, rocks will get moved around and turned over and stuff. So if, if you see a bare rock next to a rock covered in seaweed, it could be that the bare rock was recently moved to that location by the ocean. But I see that phenomena on bedrock too, which isn't moving around a lot, and that's just as mysterious to me as it is to you. And I think you would be hard pressed to find someone who can give you a definitive answer on that, because seaweeds are mysterious.
So yeah, a lot more thanks for your amazing presentation, as you can probably read yourself in the chat box. Everyone has loved it so much. And then I noticed Amanda, your colleague, has said that if anyone has questions about the seaweed class at Eagle Hill, to send her an email, which is provided in the chat box, posted at 4:59 if anyone wants to go back and look at that. And then it looks like we have a question here, which you kind of, I think, answered, but in a way, do you ever help with seaweed IDs on iNaturalist? And because this person doesn't always get a confirmation.
Yeah, I do. I haven't gone through it in a month or two, so if you posted recently, I probably haven't. But I do enjoy taking some time here and there to go through and add species determinations. I've never posted my own data because I have different systems that aren't really conducive to iNaturalist's format. But I think my name on there is like Jordan Seaweed or something like that. But if you go through any post from like the last year or so, you'll be able to find me on there. And the only time I don't give an identification is when there truly just aren't enough features in the photograph for me to even give a guess.
One of the last questions for those of us who are new to Maine and eager to explore for macroalgae, where would you recommend that we head at present? Down east, Pembroke, Mount Desert Island?
If you're just looking, there are some beautiful places in Acadia, like Ship Harbor on the southwestern side of Mount Desert Island has great seaweed diversity. Same with Seawall, also on the southwest side. Otter Cliffs area is sort of dangerous, so I don't recommend doing anything sketchy, but usually places that are dangerous because they have a lot of wave action and easterly exposure are the places where seaweed is the most happy and diverse. So use good judgment, but try and head for places that are like peninsulas and rocky coast rather than sandy, because seaweeds need, stable substrate to anchor to. But there is no harvesting permitted in Acadia National Park. You should always check with the landowners or land managers wherever you're exploring before you harvest anything, whether that's a place like Blue Hill Heritage Trust or private property or Acadia. It's good to cross your T's and dot your I's in that regard.
Yeah, thank you, Jordan. Somebody's wondering if you could reshare your email. Is it the COA email?
Yeah, I'm putting it in the chat right now.
Okay. Cool, thank you. And then kind of in a similar vein, someone was wondering if you could share how to submit photographs to you.
Email me. Yep, same email address. Oh yeah, there's a comment about invasive seaweeds, and that is loaded terminology, right? Like, what even is invasive? And my definition of invasive is a plant that is disturbing, a recently arrived plant that is excluding other species and creating a monoculture. So there are tons of introduced seaweeds that I would not call invasive. They're just here, they kind of exist and do their things. They're not displacing other things, but there are a handful that are truly invasive. They create monocultures and exclude other species. And Dasisiphonia japonica is definitely the most voracious one that I've encountered and the one that I'm most concerned about. But it's interesting that there isn't really any official entity that is delving out designations on these introduced seaweeds and like what our concern levels are on them. There's very little kind of official literature out there about that stuff, which is why I am interested to get part of the reason I went over Dasyphonia japonica is I want people to be able to recognize that one, and I'm interested to know where you're finding it and when you're finding it.
Well, thank you so much, Jordan, and thanks for
Yeah,
staying on a little later too, an extra 10 minutes to answer questions.
Mhm.
thank
We're
you guys.
yeah.
Yeah, I really I'm blown away by the number of folks who showed up to learn about seaweed, and all of your positive comments and encouragement are so appreciated. And I really look forward to sharing the guide with you.
That's wonderful. Someone asks how many of us are here. At a certain point, there was over 80.
Mhm.
So really wonderful turnout. Thank you so much, Jordan. Keep following your passion and sharing your passion with the world. It's absolutely
Yeah.
contagious.
Mhm. I can't wait for the field
Feel
guide.
free to email me with all of your CBD news. Awesome. All right, guys.
You have
Thanks again.
Bye.
bye everybody.
Bye.